Hello everyone, welcome back to our channel Biology Reader. Our today's topic is plant growth regulators, also known as phytohormones. In today's video, we will discuss what are plant growth regulators and their application in plants. So friends, let's begin. What are plant growth regulators? Plants rely on a group of chemical substances known as plant growth hormones to signal, regulate and control their growth. Plant growth hormones are the chemicals that plants naturally produce to control the length and thickness of the plant. These are also termed as phytohormones or plant growth regulators. Plant hormones regulates the physiological activity of plants by either stimulating or terminating plant maturation. So in simple, we can say that plant hormones control the growth behavior of plants. PGRs can be found naturally, known as phytohormones or plant hormones, or can be derived synthetically, known as plant growth regulators. Based on the action of phytohormones on the growth behavior of plants, these are divided into two categories: plant growth promoters and plant growth inhibitors. Hormones that help in growth of plants are known as promoters. In contrast, hormones that restrict the growth process are known as inhibitors. Auxin, gibberellins, and cytokinins are kept under class plant growth promoters. Abscisic acid and ethylene are kept under plant growth inhibitors. Friends, one thing to note is that ethylene can be kept under both classes or categories because it acts as both promoters and inhibitors. Still, its inhibiting features are more than promoting features, that's why it is generally kept under inhibitors. Now, let's understand each hormone in detail along with their function. First one is auxin. The primary naturally occurring auxin in plants is indole 3-acetic acid. It has a simple structure consisting of an indole ring and carboxylic acid group. Auxin is produced from the apical region of the stem and roots. It was the first phytohormone to be discovered among all. The term auxin means to grow. Auxin can be derived both naturally and from synthetic sources. Indole 3-acetic acid and butyric acid are obtained from the natural sources, while naphthalene acetic acid and 2,4-dichlorophenoxyacetic acid are obtained from artificial sources. Function of auxin. Auxin promotes flowering in plants. It is also widely used as herbicide. Auxin prevents dropping of fruits and leaves in early stage. Auxin mediates phototropism in plants. It is also used for food production by parthenocarpy. Auxin helps to initiate rooting in stem cutting. Next one is gibberellin. Gibberellins are a group of over 100 naturally occurring compounds. The most known gibberellin is gibberellic acid. The basic structure of gibberellins consists of gibberellin ring system. Gibberellins are produced from young tissue of shoots in developing seeds, leaves and roots. Function of gibberellins. Gibberellin induces maleness in dioecious flowers. It also produces alpha amylase in germinating cereals. It delays leaf and fruit senescence. Gibberellin breaks seed dormancy so that it can germinate into a plant towards the presence of light. It also helps in growth of parthenocarpic fruits. Gibberellin stimulates stem elongation. Cytokinin. Cytokinins are adenine derivatives, which means they are derived from organic base adenine. One of the natural occurring cytokinin is zeatin. These are produced in cell dividing regions like roots and shoots. Function of cytokinin. Cytokinin induces apoptosis at high concentration. It also enhances stomatal opening in some plants. Cytokinin helps in chlorophyll synthesis. It stimulates cell division. It also promotes morphogenesis in tissue culture. Cytokinin stimulates the growth of lateral bud and leaf expansion by cell division and enlargement. Ethylene. Ethylene is a simple hydrocarbon gas with double bond between two carbon atoms. It is synthesized by most plant organs including ripening fruits and aging tissues. Function of ethylene. Ethylene induces shoot and root growth. It also induces femaleness in dioecious flowers. Ethylene stimulates the phase of dormancy in seeds. Ethylene promotes abscission of leaves and fruits. It also promotes fruit ripening. Ethylene helps in flower opening. Abscisic acid. Abscisic acid is a simple small molecule consisting of a carboxylic acid group and branched isoprene chain. It is also known as stress hormone as it helps by increasing the tolerance of plants to different kinds of stress. Function of abscisic acid. It stimulates the closing of stomata in epidermis. It also helps in development and maturation of seeds. It inhibits plant metabolism and seed germination. It also inhibits shoot growth. Abscisic acid inhibits the activity of alpha amylase stimulated by gibberellins. It makes plants resistant to water deficiency. Friends, we will end up this video by application of plant growth regulators. Germination. Germination is the process when a seed begins to grow from the embryo of dormant seed. The application of auxin and gibberellin breaks the dormancy phase into metabolic phase. Dormancy is a resting phase where all metabolic activities are suspended. Rooting. Auxin is a rooting hormone that is widely used on stem cuttings. Callus induction. Cytokinin and auxin induce callus formation from the explant in tissue culture. Growth inhibitors. PGRs inhibits apical growth and promotes lateral bud growth, which results in bushy plants with an increased number of flowers and fruits. Dissecant. PGRs artificially promotes the drying of plant tissue. It is a hasten drying process that results in the removal of leaves as it eliminates water from green foliage. Fruit ripener. These are the substances that can fasten the ripening process and artificially ripen fruits for commercial purposes. So friends, that's all for today. Today we have learned about some organic molecules that perform their regulatory functions for the growth and development of plants. If you find this video helpful, do like, share and subscribe to our YouTube channel and don't forget to press the bell icon for more videos.